Okay, come on. So we left off last time talking about some of the cells. This table distills down truly a wealth of information. A lot of stuff going on here. So I'm not going to go through and spout off all of this stuff, but I will give you a little bit of tidbits as we move through here. There are a few slides after this that blow these guys up and give you some extra details as well. So I'm not going to just get bogged down right here. So we talked about, back it up here, the hematopoietic stem cells that have the potentials to give rise to everything else down below. There are numerous mechanisms at play that stimulate these cells, or those that have started the initial differentiation, different cytokines that stimulate the production of the respective cells that we see at the bottoms of all of the arrows, all right? Which is what we've got right here. So the table puts it into a bit of perspective, giving you some numbers as far as the quantities or percentages of these players. In our circulation, it makes sense that the majority of the cells are going to be erythrocytes or red blood cells, boatloads of them. And we're representing all of these in a microliter quantity, so a very small drop, all right? 5.2 million erythrocytes in that little drop of blood, massive amounts. And if you were to look at a blood smear under the microscope, that's what you're really going to be seeing predominantly are erythrocytes all over the place. Little kind of pinkish colored donut shaped cells all over, all right? Erythrocytes are kind of special. These are cells that don't have a nucleus, all right? And that kind of gives them that sort of puckered in or jelly-filled donut sort of shape, because it's not just an open hole, all right? That also becomes important with some details with immunity and blood transfusions and things like that. We're not going to touch on that right now, all right? But the fact they don't have a nucleus truly is important with that. So erythrocytes, dominant players in that little droplet of blood. And then we think about the white blood cells or leukocytes. There are numerous players that we can potentially see, and also can depend on circumstances in the individual. If we're talking just a healthy individual, that's what we're looking at right here, normal ranges. But if we're thinking some situation where there are some sort of microbes causing infection, the ratios that we see here are oftentimes going to be different than the norm. And we may see elevated levels. We also can see depleted levels, just depends on some of those circumstances. So deviations from the norm sets the stage for questioning situations. Some of them kind of can serve like a beacon. If we recognize something like excessive amounts of eosinophils, what we see right down here below, that may indicate to the clinician that this patient potentially is infected with a parasite. Oftentimes, parasitic infections have very high levels of eosinophils, but proper talking with the patient, getting patient history stuff is important, because there are other situations where we may see elevated levels, and it could be like an allergy where eosinophils could be high as well. So it just kind of depends. You definitely see more of that with basophils. They can be more common in an allergic sort of reaction. Same with the mast cells. We mentioned a little of that last time. So again, percentage-wise, getting off topic with that a little bit. With the white blood cells, the first bit that we look at are those granulocytes. Remember, different enzymes and acids within those lysosomes, and different staining characteristics or qualities with the common, Wright or Giemsa stain. They show up different, all right? Neutral, acidic, and basic staining, respectively. So the main players with the leukocytes are going to be the neutrophils. They're the dominant cells. I'm looking at a blood smear. Oftentimes, that's really what you start seeing a tremendous amount of. Lots of these weird-looking nucleated cells. I mentioned before, we oftentimes describe these as PMN cells or polymorphonuclear cells because of the odd-shaped nucleus. It is just a single nucleus, but it looks like multiple nuclei within there, all right? So between 50 to 70% of the leukocytes are going to be these neutrophils. A small fraction of really what we're thinking about, few thousand, 4,000, maybe a little bit more, compared to the millions that we're seeing with the erythrocytes, thousands, all right? And total, all of these leukocytes, just a little bit more, 4,300 or something like that, right? Fluctuating a bit, dependent upon some situations, all right? Eosinophils, much less, and all of these considerably lower as we look at this. 1 to 3% of the cells in that microliter of blood. Basophils, less than that, maybe 44 or so. Takes a long time of scanning fields of view with the microscope, patience, perseverance to truly find either of these, really, all right? The next players are the agranulocytes. There are handfuls of players in this category, lymphocytes and monocytes, 2,600 cells with these guys in that microliter. So the lymphocytes, these are T cells, B cells, that's these littler bits, and some natural killer cells, bigger ones, all right? T and B cells, looking at them in a blood smear, you can't tell the difference. They look basically the same. You can't say which one you're looking at, all right? And then lastly, the monocytes. I mentioned with these, they will move out of the circulation into the tissues and differentiate into macrophages or dendritic cells, either way. We're not looking at that right now. We're only looking at what's in that droplet of blood, right? You don't see macrophages or dendritic cells presented up here because that's not where we see them, all right? And then lastly, the cell fragments, those platelets, all right? A category in and of itself, all right? And a couple hundred, 350,000 cells or more, all right? And these important, like I said, in wound healing, stimulating mitosis to progress. So lots of different activities with these cells, but truly the idea is immunity, surveillance, looking for foreign invaders and coming up with some sort of strategies. Some of these cells, like our neutrophils, are what we describe as phagocytic cells. The same holds true to those dendritic and macrophage cells. They've got the ability to seek and engulf some foreign invader, destroying it, all right? Neutrophils, I should skip ahead because I know there's more details with them. So the neutrophils, here we can see an actual smear, what they look like, and then the cartoon image below. So lavender, pinkish, orange sort of staining, and deep blue granules within the cytoplasm. And you can see the odd looking nuclei, all right? So with the neutrophil being a kind of main player when we think about something maybe getting into the circulation, these cells phagocytize, destroy those foreign invaders, and ultimately in the process of doing this, they're going to die too. They basically are being produced pretty rapidly, and in a bout of infection, they're really being destroyed very rapidly as well. A lot of pus that you may see in a wound, a lot of that are dead neutrophils. There are also dead organisms and some of our own cells within that as well. But a lot of it, it's it's the neutrophil. All right. When they die, when they basically lyse after they have got this foreign invader, they spew out all sorts of material, lots of DNA within their nuclei that is really kind of like a fishing net that gets cast out with the hope that all of this debris snags other combatants, other microbes, whoever they are, and traps them within this net, providing an opportunity to slow down those microbes and give other cells a chance to pick them out of that netting that they have released. A pretty cool strategy, all right? Eosinophils and basophils don't really, although maybe there might be a little bit of phagocytosis, but I think for the most part, these cells aren't phagocytic cells. They do extracellular killing. So they see some invader and they don't engulf it, but they dump their contents to the outside and try to do the damage extracellularly. And in circumstances like a parasitic infection, back to the eosinophil, if we're thinking about some sort of protozoal organism, they're typically going to be quite a bit bigger, or bigger for sure, than these eosinophils. So trying to pull in and phagocytize something that's bigger than you isn't going to happen, all right? So dumping contents onto the surface of that thing is a strategy that they employ, all right? So we'll get a little bit more with some of that as we progress along. So mast cells, like we mentioned before, and as we say right up here again, have similar roles like a basophil. And a lot of the idea with the mast cell is to release chemical mediators, more cytokines. Histamine specifically is a very good one that these cells, along with the basophils, get stimulated to release. And in an allergic reaction, which is where these mast cells really shine, if you will, they dump mass quantities of the histamine, which in turn stimulates some of the symptoms that you exhibit during an allergic reaction, hence taking an antihistamine to block this activity. All right. So that's what we're seeing here in the blood smear. But we also can see an image over here, a tissue section, where the mast cells have the opportunity to migrate out of the circulation into the tissues, like a monocyte moving out into the tissues and becoming a macrophage, all right? So the mast cells have potentials to work outside of the circulation, all right? There are other cells, neutrophils, can also move through the vessels and get into the tissues if we're thinking some sort of wound. Pretty sure the other video I posted talks about the same deal, a wound on the thumb, and goes through the whole process of what's happening, and you can see multiple cells like those neutrophils showing up. So natural killer cells, these cells are doing surveillance, and with a name like natural killer, I think it's pretty obvious what they want to do. All right. They would like to destroy anything that they recognize as being a little bit off, if you will. All right. So these cells really have more of a responsibility of surveillance of our own cells is really kind of the idea with this. So here's our natural killer. And here are cells that have specific proteins on their surfaces that are like a molecular fingerprint. All right. We call these MHC1s. It stands for major histocompatibility complex. And it's our own cells' molecular signature. All right. If you donate not blood, because these aren't participating in that, but if you're donating organs, tissues, whatever it might be, bone marrow, they want to know your blood type. They need that information, but they also want more information. They want details about your MHC1. And trying to find a close match isn't saying this person's type A positive, you're type A positive, you're going to match. Just because you're the same blood type doesn't matter. All right. Could for a blood transfusion, but not bone marrow. All right. If your MHC1s are close matches, now you're a potential candidate. All right. So MHC1 complexes are found on all of our cells that are nucleated. They're not found on the red blood cells. And that's how blood transfusions can work, because we don't have these complexes to be recognized as that's not mine. All our body sees is you're type A, you got injected with type A blood, you're good to go, right? Organ donation is where it becomes a problem, all right? So these cells, our own cells, have these fingerprints, have these complexes on the outside that say, this is me, leave me alone, all right? And the natural killer cells have receptors on them that basically recognize our own specific fingerprint. And these cells say, I know you're good to go. I see what you've got. You're my own, all right? And they leave them alone, all right? This is a healthy situation, all right? They become important, the natural killer cells, with cells that become infected, virally infected cells, or even cancerous cells can be targeted, because cancerous cells oftentimes don't exhibit the stuff they should be exhibiting on the surface of their cells. And the natural killer cells recognize that. So in this case, the virus infects this cell. Now we're no longer a normal, healthy cell. We're not exhibiting this proteins on our surface like we should be, or those proteins are there but not shaped properly, so we can't fit like a lock and key, and nothing there for our inhibitor to recognize. So it gets activated. It binds to the cell, different protein, allowing the docking of this. But when this occurs, this cell gets destroyed. Apoptosis, again, cell suicide. All right. It causes it to get destroyed. And these natural killer cells don't need to be primed by anything. They don't need to have some antibody say, look at these bad guys. They're looking at this stuff all of the time, looking at our own stuff. All right. Trying to find bad news. All right. And if they happen upon that, those cells get quickly deactivated. All right. So natural killers. And back to this, that's these guys right here. All right. So one of the mechanisms that they utilize, and it's not exactly the same, but it's pretty much the same idea as that membrane attack complex that complement proteins are doing. The natural killer cells release some proteins, perforin granules, that basically bore holes in the cell, but in our own cells, right? Causing those cells to lyse. So here we've got another blood smear. Lots of different players up in here. a few pointed out, erythrocytes are all of these guys. All right. All of these are the red blood cells. And like I said a minute ago, it's going to be the majority of stuff you're seeing on the smear in the first place. Far more red cells than anybody else. All right. So one neutrophil pointed out, and then a handful of monocytes, more of them that aren't being shown either. All right. All of these are going to be monocytes. And I mentioned, I think this was last time, the monocytes typically almost twice the size of the other white blood cells. We see a neutrophil and then this guy right up above. These are giants compared to our neutrophils. Neutrophil's a little bit bigger than a red cell, all right? And these guys quite a bit, all right? So all of these are monocytes. This guy right here, likely lymphocyte, all right? T or B cell. That's my gut saying that. What's this guy? Super little, looks nucleated, but it's not. This is a platelet, all right? So little, considerably smaller, little fragments, but still some hints of what looks like maybe some nucleus, part of a nuclear membrane likely in these little fragments, but it's not nucleated, all right? So the monocytes in our circulation, moving out of the circulation into the tissues, and now we're thinking macrophages. That's what we're seeing here. Handfuls of macrophages, all nucleated cells that we're seeing right here. And in the tissues, their job is, again, surveillance, looking for stuff like bacteria or other microbes and phagocytizing. Lots of stuff with macrophages. They are super good at finding these things and destroying them, but they also like to tell everybody what they've done. And once they have pulled something in and destroyed it, it presents little fragments of those microbes on the surface. And then we classify these macrophages as antigen presenting cells. I just found somebody, I destroyed them. Now it's time for show and tell. And I want to get everybody up in here and I want to show them what I did. And ultimately, somebody, little T cell, will recognize it and tell somebody else. That's the hopes once we become this antigen presenting cell, to keep the process of finding and destroying these foreign invaders moving, all right? So we'll get to some more of that in a little bit. So this image, very similar to the next video with a wound and all sorts of details that are happening. So we have some situation. The barrier of the skin gets broken, you scrape your knee, you cut your finger, whatever it is. And once that barrier is broken, the potential for something to become inoculated through that wound is there. So it could be something that was on the surface of your skin that's now able to get in, or it could be something that was on whatever it was that cut you, right? Something that's able to get into that wound, all right? Maybe a scrape, and where the scrape happened, you had some Staph aureus, and the Staph aureus now gets in. And now it's in a location where it's got ample nutrients instead of a very desert-like environment. And these guys are like, sweet, look at all this food, right? Whatever. Many stories you could think about with this. So whatever, the green guys, little bacteria, these are the pathogens, and they make their way in through that wound, all right? There's a lot of stuff that starts happening after the wound happens. There is going to be numerous cells trying to respond to this, and there's also going to be other cells that are important players in stopping the blood and starting the healing process. Platelets play a big role in that, all right? So as this happens, all right, the pathogens get in, and as they make their way into the tissues, all sorts of chemical messages start being released. messages that the bacteria or the pathogens harbor on their outside surface can be picked up, and other cells, our own, start releasing other chemical messengers to call in the troops. All right. So these signals are released out as the damage happens, right? Something has occurred, and these wounded cells start releasing molecules. Cells start trying to respond. Macrophages moving through the tissues recognize, bump into, and engulf, phagocytize those foreign invaders. And in the process of doing this, they start dumping out chemical messengers, trying to call in more troops. All right. So pro-inflammatory molecules and chemotactic cytokines, beacons, basically saying, come to me, come see what we have here, right? Inflammation is kind of a double-edged sword, and a little bit of inflammation helps to stimulate some of these cells. Excessive amounts of inflammation can truly cause issues in the host, all right? And hence the plethora of anti-inflammatory drugs. So as all of this happens and we start getting chemical messengers just secreted, the blood flow increases. And as we see some of this with the inflammation, some of the white blood cells are able to wiggle through this. We call it diapedesis when they are moving through the cells into the tissues. So monocytes moving in and differentiating into macrophages, neutrophils moving through there with phagocytic properties and the potential to destroy themselves with their gigantic nets that they release out. All of these things start showing up, trying to combat it. Up here, these are complement proteins. Complement proteins also are stimulated. They're moving out of circulation. 15, what did they say, quintillion or something like that? That's a massive amount of complement proteins. Impressive, all right? Then it makes me stop and say, how the hell do they know that, right? Who counted them, right? That's a lot of proteins, all right? So as all of this starts happening, you can see here the neutrophils and their releasing of cytokines and degradation, all right? And truly mopping this up. We're not completely done with it yet, but we've stimulated these cells to come in and recognize, all right? There's going to be more stuff happening, all right? So there are lots of things that I think about that really are like those MHC ones that we think about on the surface of our cells, our own cellular fingerprints, all right? White blood cells have some other markers on them, and microbes have other markers on them, all right? Fingerprints on everybody that allows us the opportunity to do some detecting, all right? To see, is this my own stuff or is it somebody else? And if it's somebody else, we better do something about it, all right? So this is a phagocytic cell, all right? And all of this other stuff are things that we could potentially be recognizing, all right? So the phagocytic cells, the macrophages and dendritic cells, and our neutrophils, they've got specific receptors, these green guys, and technically there's some others. This one was outside before, but we've pulled it in a vesicle. Once we've linked up with that receptor, we cause the cell to invaginate, pulling that in a vacuole, and then we'll destroy, all right? So this phagocytome, we've already phagocytized this, all right? So on the surface of the phagocytic cells, are these PRRs, pattern recognition receptors. These are things that are recognizing the foreign stuff, all right? On the microbes, they've got different things, their own fingerprints, and we call these PAMP, all right? And this is a pathogen associated, does it tell me in here? I don't remember what it is. Pathogen associated molecule. I don't know. I know it'll tell me in a second. I'll be like, I knew it, but I didn't. So many abbreviations. All right. So if we recognize any of these things on those receptors, it's going to stimulate phagocytosis, pulling them in these vesicles and the destruction. All right. So lipopeptides, these are substances that we recognize on gram-negative bacteria, the lipopolysaccharide outer membrane, all right? Some bacteria have flagella, and flagellin, proteins that make up the flagellum, are recognized by these receptors. Bacteria, doesn't matter, positives or negatives, have peptidoglycan that make up their cell walls. There's receptors for that. There's also receptors for nucleic acids. That's this one up above that we've already caught and engulfed and brought in, all right? All sorts of things that we can recognize as foreign based off of these PRRs, all right? Oh, it's right here. Pathogen-associated molecular patterns, if I would have just looked a little bit closer, all right? Reading, okay. So, with phagocytosis, our big blue guy up here is a macrophage, all right? And this macrophage, in this case, has recognized a bacterium, our brown guy in here, and something on the surface of that cell, lipopolysaccharide, flagella, whatever, all right? We've seen something, and we pull it in, all right? As we engulf that bacterium and pop it in, it's now in a vesicle, and we call that a phagocytome, all right? That phagocytome, after it has been brought into this cell, is going to fuse with a lysosome. And this becomes a phagolysosome, all right? It's the two together. If we aren't really thinking about white blood cells and all of this stuff, sometimes we call it a secondary food vacuole. After we've engulfed some food stuff and then the lysosome links up with it, it's a secondary food vacuole, and we destroy it and release those nutrients out. In this case, we're not thinking about eating them, we're thinking about destroying them, right? They truly are kind of synonymous, but for our white blood cells, it's a phagolysosome, all right? With phagocytized, now we're destroying. So all of those digestive contents, enzymes and acids, all sorts of substances, boatloads of compounds are found within the lysosome. They all, in the process of producing these molecules, coded for by specific DNA, we go through translation in the rough endoplasmic reticulum. We bleb off a little bit of that moves to Golgi, and Golgi starts taking all of these little blebs and says, oh, this acid, that acid, these 5 enzymes, I want all of you together. And it packages up boatloads of these different substances all into one vesicle, blubbing it off of the other end of Golgi, and now you've got a lysosome filled up with all of the ingredients it needs to destroy, all right? So these lysosomes make contact with the phagosome, fusing and dumping all of their digestive juices, if you will, over the bacterium, destroying it, all right? And now we see taking out the trash, some of the undigested garbage gets pushed to the outside, all right? We're not showing antigen presentation right now. There's other stuff that happens with that, all right? And in those circumstances, little bits that aren't going to just be the trash they're going to be shown on the outside of this macrophage. So these little fragments associate with MHC proteins. And in this case, these different than our own cellular fingerprints, right? And it puts that with that protein on the outside. Now it's showing what's up. It's been, it's got ornaments on it. It's been bedazzled, all right? If you remember those commercials, right? Bedazzling everything, right? Look at my jeans. They're bedazzled, right? If you don't know what I'm talking about, I apologize. All right, but that's what they do, all right? Some of you you're like, I'm bedazzled right now, all right? Okay. We'll see more with phagocytosis and really that antigen presentation, because that's more important truly for the stimulation of antibody production. And that's the next chapter. All right. So just hold tight. You're going to get more of it. This image, I like it and I hate it at the same time. I've got a few problems with it, but it's okay. I'm going to deal with it. All right. You'll know my problems in 1 second. So leishmaniasis, this is a type of parasite. All right. Leishmaniasis has potentials to exist in two different types, and there's different types of Leishmania parasites that exhibit these characteristics. Some of these parasites result in skin infections, cutaneous leishmaniasis, and other parasites result in a visceral form. So it's multi-organ involvement. Deep down, I think if you want it, you probably want the skin one. All right. You don't want either of them, truly. All right. So the parasites are pretty impressive. These cells get into macrophages and survive and replicate and progress to new cells, living in the cell that should be killing it. Awesome. It's a brilliant strategy for a pathogen, parasite, whatever, all right, to live right inside of that cell. They hijack the machinery and prevent, the lysosomes from doing the killing. And it gives them this house to replicate in. Nobody's going to know that these bad guys are in them, because the cell that should be destroying them is the one that's got the bad guys in it, right? What are you going to do? Nothing, right? You can't do anything about it because there's nobody else to deal with them, right? Kind of crazy. So multiple things. I'm not going to go through all of the details of this. We will talk a little bit about Leishmania later on when we think some infectious disease. But my problem with this is the picture, because this guy looks as if he's got cutaneous leishmaniasis, but he doesn't. All right. And I only know that because I teach parasitology and I've seen many of these pictures. I've never seen true instances of it, but I know he's got something else going on. All right. And what he has, yes, it's Leishmania. All right. But what he is exhibiting isn't the cutaneous form of this parasite. All right. What he has is the visceral form of the parasite. He's got other problems going on, liver, spleen, all sorts of issues with these organs, all right? And this problem that we're seeing on this guy, we call it post-Kala-Azar dermal leishmanoid. Huge mouthful. And what it is he has either stopped taking his antiparasitic drugs, or he isn't taking them properly. He doesn't have the right dosage, something like that. When the drugs aren't taken properly, it causes these skin manifestations to appear, all right? So it looks super disturbing. And to just say, oh, this is Leishmania makes you think, holy cow, I never want to get it, which you don't in the first place. But really, cutaneous leishmaniasis is like a little ulcer, a little scab that you see on your skin. Nothing like this, all right? And if he gets the proper dosage or he completes the stuff like he truly should have, this skin manifestation will go away, all right? So it's not exactly the same, but the picture is disturbing, right? So sometimes issues. I was scrolling through newsfeed and saw something about syphilis being very prominent. I saw that for Idaho just recently, but the one I saw before was talking about in Utah. And the image that they showed wasn't syphilis, but how would you know, right? You have to be sitting in this class probably to know. The pictures that they were showing were chlamydia, not syphilis. All right. Syphilis is a little corkscrew spirochete. All right. Chlamydia, little bacteria, not corkscrewed, a bacterium that is an obligate intracellular parasite. It's got to get into your cells. And when it does, those cells look very different. We call them McCoy cells. That's what the pictures were, these dark brown looking cells with a bunch of other cells. And they're like, look at this syphilis, right? And if you don't know anything about it, you'd be like, holy cow, that's disturbing, right? But that's not the picture, right? That really is syphilis, all right? Another one that I'm just like, come on, right? But if you don't know, you just accept that, right? And then you're thinking that's what syphilis looks like, but it's not, right? Okay, enough of that. And oh, enough of that. Holy cow. Farewell, good day. It's a good thing I looked at my watch. I'm ready to just keep going. I bet Dr. Meldrum's out there wondering, what's up with Shirley? All right. Farewell, good day.